I think let's go ahead and roll to be respectful of people's time. So what we're going to go through today is a presentation that I've been giving for a couple years on organisms of Lake Merritt, predominantly looking at the invertebrates, because they're the things in the kind of the largest numbers that are also the kind of wackiest and wildest things there. They really give you a feeling for the amazing amount of life that is in Lake Merritt that's constantly mixed. Let me give you a little bit of background though on some things that are important, I think, especially tonight. And then we'll drop into kind of the main meat of the presentation. So I'm going to share my screen. So you guys should be able to see that first slide now that says beneath the surface of Lake Merritt. Myself and Constance, who you've heard speaking in the background there, and Tony, who's also on this Zoom meeting, we're part of the California Center for Natural History at calnature.org. We set up this presentation actually far in advance before a lot of the things currently going on tonight, and we wanted to give those recognition that here in downtown Oakland tonight, there is a sit-in protest taking place at 14th and Broadway. So we know that a lot of people cannot join tonight because they are going to be down there involved in the protest. If you feel compelled, there are a number of organizations that are doing good work around these protests that are local to the Bay Area. We're highlighting 3 here in the slide deck. There also will be links in the chat. And so if you feel moved by this presentation, please consider donating to one of these groups. calnature.org, California Center for Natural History, we're a nonprofit that does a lot of, I would say, like outdoor education, especially for urban adults, but also with social justice mixed in. And so at this point in time, it is most important, we believe, to support these groups that are supporting what's going on locally, and it affects globally kind of what's happening in this realm. As we're making our way through this slide deck today, if you come up with questions, if you place those into the chat, we're going to basically kind of pool those together at the end and then do a Q&A at the end of the slide deck. And Constance, if something comes out that we need to talk about right away, just feel free to kind of butt in. I
know I do.
I know, just want to make sure you feel comfortable. So let's get started with thinking about what Lake Merritt is, first of all, because to understand the organisms in there, we have to understand a little bit of the history and the context around this place. So for those of you guys that are not from Oakland, Lake Merritt is often called the jewel of the city. It sits right in the center of the city. It's a major park that has an extreme focus for communities throughout Oakland. It, though, is not a lake. It is actually a tidal lagoon that was formed after the last ice age. Human settlers have been there for at least since 4000 BC. The Hokan, we believe, are the first people that were there, and we believe they were there about 4000 BCE. And this is based upon shell mound dating in Emeryville. There were also shell mounds around Lake Merritt as well. Eventually, the groups that were there when Europeans showed up are the Ohlone and the Costanoans that probably came in a lot closer to 600 CE. When we get to kind of the European influence on the lake, the history is, I'll kind of be quick about this. For the most part, it was part of this large land grant by Luis Peralta, the last Spanish governor of California. He divided basically all this land amongst his 4 sons. In fact, at the time, the lake was named after Luis Peralta. 1849, of course, California is annexed by the United States. So all of a sudden, there's all of this pressure on this land by foreigners coming in and basically squatting. So between squatting and selling, Louis' sons basically lose most of that land by 1897. 1853 was when the first toll bridge was built across San Antonio Creek, kind of where 12th Street is right now. And what that did was it connected downtown Oakland with the other portion, the other side of the lake, which was known as Brooklyn at the time. 1860, the population of Oakland was about 10,000 people. And in about 10 years, by the time we get to 1870, that population gets up to 100,000 people. So you can imagine the influence that has on basically waterways coming down into that Lake Merritt Basin. It is huge. The lake itself derives its current name from Samuel Merritt, who's the 13th mayor of Oakland. He was this giant, imposing character of about 6'3", 340 pounds. He was a big portion of the Oakland Waterfront Company, basically a monopoly of Oakland's kind of shorelines. It is under his mayorship that the main dam goes in at 12th Street. And in 1870 is kind of one of the things that we should actually kind of really be aware of right now this year, because we're actually celebrating its 150th anniversary, which is basically Lake Merritt being designated as the first wildlife refuge in the United States. And this was partly due to Merritt's kind of pushing on Henry Hay at the time, the governor of California. And the thing to think about there is that this precedes the national park movement by 2 years. And who was living over the hill from basically Oakland at the time in Martinez? This weird lanky guy named John Muir. And so I'm sure that this had some effect on John Muir's imagination as far as what may happen with other natural landscapes across California and the United States. Rumor has it though, Merritt wasn't doing this entirely out of like goodwill, but mainly because his windows were getting shot out a whole lot because people would go down to the lake and shoot birds, because a lot of birds go down there. And also his neighbor's cow had been shot. And so, you know, a little bit of personal interest built in, but it ended up being something good for all of us in the long run. 1874 is when we first start hearing the lake called Merritt's Lake, and it soon kind of gets derived and moved around to Lake Merritt. Remember, we had that huge population boom. Basically, 90% of that population human waste in 1884 is flowing down from kind of the Oakland Hills and the shallow hillsides around Lake Merritt down into the lake. In 1892, city engineers report that there's a sedimentation rate of 1 inch per year. I'll let your imagination tell you what that material is that is sedimenting the lake when there's that many people living close by. 1893, we actually start dredging Lake Merritt, and this goes on for about 20 plus years. Material from that dredging, basically a lot of human excrement, ends up helping fill in more of that 12th Street Dam. It's used to build part of the Bird Islands Refuge. And then in kind of 1907, 1925, we get these city beautification projects, which then kind of double down on the majesticness of Lake Merritt and get you to forget about all of the human waste in there. So you've got pretty postcards that are nicely stylized and colored. You've got people swimming in the lake, even though the waste streams are still going in there kind of full on. These are just old historical photographs I usually show when I'm talking to classes, just so they have an idea of what Oakland looked like at the time. This 1900 one, I think, is a really dynamic one because it gives you an impression of urban density around Lake Merritt. Adams Point, not too much going on. Downtown Oakland, fair amount going on. Brooklyn, slightly developed. And then you also see that large fill down where 12th Street is, where Lake Merritt's draining out towards the estuary and the bay. We get this big tide change, though, I'd say in the 80s, when city engineers finally decide that, hey, maybe all that sewer stuff shouldn't be going down into Lake Merritt, and basically reroute a bunch of that to our water treatment plants. I would say the most notable thing, at least for me in my time period, is Oakland passes Measure DD in 2002. And this is almost a $200 million bond that works on more or less cleaning up the inputs coming into Lake Merritt, and that's the 62 storm drains, the 8 creek flows, basically that 4,500 plus acre watershed that's draining down into Lake Merritt. It also worked at widening the channel, more or less getting rid of some of that fill that had taken place early on 100 years ago in order to get more tidal flow with the bay taking place in Lake Merritt. And there's a whole bunch more that goes on with that. You can learn more about that at waterfrontaction.org/DD. My first introduction to any of this is actually in this photograph right here. That guy in the top hand right, his name is Joel Peters, and actually Constance Taylor, whose voice you've been hearing, is the person that kind of put on this walk. And I, at the time, was just somebody living in downtown Oakland, kind of thought Lake Merritt was just like what everybody else thinks, which is that it's probably just a gross body of water full of a whole bunch of poo and things you'd never want to touch. And this one walk kind of piqued my interest. And I started thinking, like, there's a lot more going on here than I can see by just walking by every day. Maybe I need to start investigating at a deeper level. And so thank you, Constance, for that walk years ago. And thank you all those people that worked on Measure DD. What you've done, I think, has really changed what I've seen over the years at Lake Merritt. Another historical piece we should be recognizing here is that the entire study of marine invasive species basically started at Lake Merritt in the 1960s. There was a high school kid by the name of Jim Carlton who was collecting organisms from the lake and then trying to identify them. And he was running into this problem where he would collect things and he'd bring them to like Berkeley and places like that, and they couldn't recognize them as something from here. And at some point, Jim sends stuff off to Australia because he saw something in a magazine, and it comes back that one of the researchers there is like, oh yeah, that's our critter. And this kind of blows everyone's mind, because at the time, everyone thought because the oceans are more or less all connected, that if something really wanted to get from, say, here to here, it could do it. There was no, quote unquote, obstacles. And Jim's kind of research, which he's still doing now, the study of marine invasives, really kind of uncovers this whole dynamic of how these organisms are prevented, A, from getting from place to place due to pelagic waters and things like that, but then how ultimately some of them get from place to place due to ballast water and boats and things like that. So Lake Merritt just happens to be a great place to study this, because if you look at that photograph down below, you've got the Oakland port, and right behind it, you've got Lake Merritt, this kind of brackish water system where you've got fresh water coming in off the hills, San Francisco Bay water coming in from the bottom. And so you've got this perfect selective environment to basically let things thrive that show up from all over the world in this ballast water that can handle a brackish water environment. And that's exactly what we see in Lake Merritt. We have critters there from every place practically that Oakland's ever traded with in the world. It's kind of amazing. Usually when I go through this with students, we do a little taxonomy thing at the start, and it used to be like really small. I'd be like, okay, these are the things that we've got here in the, phylum, animal. And now it's pretty much, we've got a representative of everything well documented in Lake Merritt. We're still working on some echinoderms, and I don't think we'll ever get a brachiopodia, but we'll get there at some point. So let me start at the bottom of like the food chain in Lake Merritt. And this is the least exciting part for everybody I know, so we'll be quick about it, and then I'll show you why it might actually be exciting. So we've got tons of photosynthetic plankton, algae, things like that going on in the lake, and a very, very large diversity of microbes. Just from a scientific point of view, this is where there is the most diversity, and we have barely scratched the surface. So this is what I used to work on at the national labs, was actually figuring out how do we look at all these unculturable organisms from these environments. And a rough estimate, we know maybe 3%, we've been able to apply a name, a species name, to maybe 3% of the organisms on the planet. And that's because the vast majority of organisms are single-celled. We haven't figured out how to grow them, so we can barely grow them. If you can barely grow them, you can rarely study them to figure out what they are. And we're just starting to be able to kind of do this stuff with advances in DNA sequencing technology. But if you dig into the lake and you throw it in the microscope, it's hours upon hours of discovery. The one thing, though, that is exciting for the common person when it comes to these organisms are some of the dinoflagellates that we see in Lake Merritt. And that's mainly not because you're going to look at them with the microscope and see how just amazingly beautiful they are, but every spring, or not every spring, every fall in kind of September or October, we'll get a bioluminescent bloom in the lake due to what we think is lignolium. I've sent this out to some groups at Santa Cruz to take a look at it. We still don't have a species name for it, but basically it tends to be brightest, it seems like, on new moons around Lake Merritt. If you go out there with a paddle and kind of brush it on the water, you'll see this big blue bioluminescent bloom take place. It's quite something to see. And on those nights as well, you will see fish jumping like nobody's business, because these organisms, what they're doing is they've kind of grown up to this large population, and then they're protecting themselves by producing this bioluminescence, because as small fish swim through or other zooplankton trying to eat them, as that disturbance takes place, you get bioluminescence that basically lights up the predator so that other predators can see it and come after it. The night that this photograph was taken, I don't think I've ever seen so many small fish jumping out of the water and having large striped bass jump out over them. We actually had fish jump into our kayak that night. It's quite a sight to be seen. So keep your eyes on walking around the lake in September, October on those new moons. So what things feed on all of these, right? If that's the baseline of that food chain, you got those photosynthetic organisms, you have the bacteria also degrading some of those guys as they fall apart. Who's feeding on all that, small stuff? Well, we have a ton of tunicates in the lake. Tunicates are a really interesting group of organisms, mainly because they look nothing like us, but besides the fish in the lake, these are the things most closely related to us. And I know everyone's kind of like going like, that thing doesn't look like it's related to me. Well, you're looking at two different stages here. The one on the left is an adult stage, and that science cover on the right is kind of the juvenile or larval stage. At the larval stage, these things have a notochord, just like you or me. They are chordates. So what they'll do is they'll basically swim around until they find a nice place with like nice temperature, some good food flow, and they'll deposit. And what they'll do is they'll go through catastrophic metamorphosis, which basically means they will break down all that notochord material, completely change their body plan to turn into this siphon system, and then set up camp to then become the mature adult. The amazing thing there is they get rid of most of that neurological network, that little notochord, that kind of mini brain they have, because it turns out brains are really only useful if you're super, super motile. And evolution, is very, very good at getting rid of excess energy use. And so it basically breaks it down, and then you have this single kind of suck and shoot system. So if you look at the left hand or the screen there, the big tube on the left, you can see is cloudy. That's actually all that zooplankton, algae, all that stuff kind of getting sucked down into this Ciona robusta. And then you can see down at the bottom, it's actually getting deposited into what looks like a little GI tract, a little colon down there, and that's exactly what's going on. You've got a little digestive chamber, and then once you're done with it, that little chute there is the outshoot, and the stuff goes out there. And so there's a whole bunch of these different types of tunicates around the lake. This specific one we think is probably from China, Taiwan, kind of Japan region. This is actually the organism, the first chordate sequenced after we did the Human Genome Project at the National Labs when I was working there, because genomes are really small and we can understand a lot about how chordates work because these organisms are easy to grow, easy to see through, things like that. We have a ton of tunicates in there. So we've got the Pacific solitary sea squirt. So this is actually one of our natives, we think. And you can find that in the lake. It's a little more kind of dappled looking. It's got speckles on its outer, it's not a shell. It's kind of like a hard encasing, because these are also weird animals. They produce cellulose, or they're convincing the symbiont to produce cellulose for them. And if you ever go to Korea, you can actually get these at most of the seafood restaurants you go to. You grill them up. They're actually pretty darn good. Don't try them from Lake Merritt, though. Filtering there, might not want to eat all of that stuff. We also have sea grapes there. This one, Mollusks manhattanensis, is one I just started to see in the last couple years. This is also, it's a relatively small tunicate, same kind of system though. You can see right through, you can see that whole GI tract, everything going on inside of there. Where you usually find most of these tunicates is on some sort of hard surface. So if you're gonna go look for these at Lake Merritt, you wanna find a hard, solid substrate in the lake. I find a lot of these on rocks or on pillars or underneath things like the boat docks.
Damon, I've got a quick question for you.
Yeah.
Since we're going through these, we had a question of what's the size? And generally, we're not going to take questions until the end, okay, but this might help put things in context.
Yeah, so the size of like Ciona robusta, it's robust. And so it can be, about yay big. I think like the largest one I found was about, I think 10 or 12 centimeters. It was an unusually large one. They're usually about half that size. Some of those other tunicates that you're seeing, though, then start shrinking down in size. Like this one that we're looking at right now, this is a whole aggregation of a whole bunch of individuals. So all of those little orange tips you see are the intake of individual tunicates, but they're living together in a colony. This entire colony is about the size of my thumb. So these are much, much smaller than Ciona robusta. These guys use a communal channel to put their excrement in, so that way you can all hang out together and not suck somebody else's poo when you're feeding, which is a great strategy in life if you're going to do colonial living. There's also other colonial tunicates that we have that are really beautiful. This is known as the golden star tunicate. And this one actually kind of comes in multiple color variants. The common name is derived from this variant, which is the most stunning one. It's the easiest one to like see. You'll be walking around Lake Merritt and you can actually just see this from the surface. These guys are relatively small. So each one of those little clusters, you know, I could fit like probably 2 or 3 of them on my thumbnail. So these guys are relatively small, but in aggregate, when you get a whole bunch of these, you can see these giant yellow, orangish mats in places. However, when temperatures change or some other environmental factor, I still haven't gotten a good grip on why this is, they will go from being that orange, yellowy color to these purple shades and totally drop out that orange, which makes them look like a completely different organism. But then if you look at the structure and everything, it's definitely the same kind of tunicates, the golden star tunicate, just in its purple form. These are a little bit harder to spot just because of their coloration, though. We also have a ton of sponges, another filter feeder in Lake Merritt. This is the most common one that you'll see in Lake Merritt, dead man's finger sponge. If you're not familiar with sponges, they're not the chemical things that were built for your kitchen. These are those are based upon real organisms. And what they do is they produce these little tiny calcium carbonate spicules that you can see in that bottom photograph that basically keep it so that other organisms don't try to eat them. They're also a really bizarre organism because they have 4 cell types, and you can more or less put a sponge in a blender and then pour it into a cup, and it will self-assemble again back into its spongy format, where it then becomes kind of a filter feeder where it can suck basically flowing water around it in, filter out food, and spit out the rest. Sponges, for the most part, get going once water temperatures in Lake Merritt kind of come up a little bit. So I've been out in the last 2 weeks and I found some really large sponges out there, larger than I'm used to seeing. So I would assume by late summer this year, we might have some really amazing sized sponges in the lake. We also have a whole bunch of arthropods in the lake that are there also eating a lot of the things these filter feeders are eating. A lot of the arthropods that get people more interested, though, are as we scale up from things you don't have to look at under a microscope. I'd say the first really cool one are these skeleton shrimp. So we have a number of skeleton shrimp in Lake Merritt. Some are from Southeast Asia, but we have one native one from California, Caprella californica. These guys can get to be about 2 centimeters, the largest one I've ever seen. And the easiest way to spot them is basically sit down at the lake, look for some sort of seaweed, and see if you see something bobbing on it. So these guys will kind of look like a praying mantis to some degree. They'll do this thing where they just kind of are rocking back and forth, usually on a piece of algae. And then when they see something go by they like, they've got those big dew claws and they'll just bam, grab it, pull it in, and then eat it. And if you look down the bottom of that organism, they've got a whole series of kind of gripping, I would call them feet for lack of a better word at this point, that they use to kind of hold on to their substrate. The way you identify this California one versus everybody else is if you can look at the back of the head, it has this weird kind of like notch sticking up off the back of the head. And it also tends to be just a lot more speckled, I've noticed, than the other ones in Lake Merritt. Other arthropods, we have a ton of these things that people would call scuds or amphipods floating around the lake. A lot of these are from Southeast Asia and Australia. Corphium is one of the most common ones you'll see in the lake. This time of year, you'll start to see them carrying eggs, which these photographs have. Those are the little kind of round pouches on the bottom of them. They've got kind of a cool thing where they'll basically swim around with these 2 arms, more or less, and then just kind of collect things in towards their mouth. So they're kind of a cool one to watch if you can catch one in a petri dish. One of the really interesting ones, I don't see it super frequently, but it's also one of these arthropods from Asia. This is predominantly from Japan, is Paranthera japonica. And it basically looks like somebody mixed a lobster with like a shrimp. It's got this long lobster-esque tail, a head with kind of these claws like some of these shrimps would have. And these guys, you know, they'll get to be about the size of your pinky or about your thumbnail. They're not huge. But if you're collecting water and just kind of like letting it sit for a while, so if you like pick up a piece of seaweed and just drop it in a dish and watch it, you'll see these guys kind of leave and then look to see if they can go someplace else. And that's usually how I spot this specific species. We also have, and this might be recognizable to a lot of you guys, because most everybody I show this to is like, oh, roly-poly. And it's like, kind of, yeah, kind of. Isopods. Isopods, roly-polies. Isopods are basically this group of organisms that for the most part are marine organisms, but everyone has contact with them because we're terrestrial organisms by finding roly-polies, pill bugs in their backyard. This specific little isopod is from New Zealand and Australia. This is the reason that no piece of wood lasts any bit of time in Lake Merritt or the San Francisco Bay. So this is an isopod that just eats the heck out of wood. So if you find a piece of wood in Lake Merritt, hold up, and what you'll look in, you'll find like these little tiny holes all over in it. And if you give it a second, we'll like, let it dry out a little bit. These guys will be like, oh crap, I'm out of my water. And they'll crawl out of those holes, and you'll see all these little tiny balls basically unravel and then walk out towards you. They are just everywhere. And these guys are exactly the same size as the roly-poly. And so you can use that as your kind of size metric when finding them. Another really cool isopod that we only have a couple observations of in the lake is this Metaceralina japonica. This one is an isopod that goes after fish. And so it actually swims around, and the only times I found it, and this is why I start wearing boots in the lake more frequently, is I was in there in a pair of sandals, and this thing swam right up to my leg and then just kept looking at my leg. And I think it was basically trying to decide whether or not I was worth latching onto. So there's a lot of these isopods that will basically swim around, find a fish, latch on, and then take little tiny bites out, and then just hang on as long as they can. Like I said, we only have a couple observations of this guy in there. You can see that species name Japonica, so also coming from kind of Japan, China, Taiwan trading routes. We also have more usual looking crustaceans, arthropods in the lake. We have the oriental shrimp, which is actually in huge numbers this year. And so this is a shrimp that people eat. These guys actually do get to be, relatively like sizable. Like you could definitely like hold one in your hand and feel the weight from it. These guys usually will be aggregating around detritus or decaying matter. So I'm usually finding these if I find rocks or I find some sort of hard substrate that had something attached to it that's now kind of in the slow decay state. These guys will usually be there. An easy way to find them at night is if you go out there with a UV light. The adults, their exoskeleton UV fluoresces, and so you can kind of hit the water and you can see these kind of UV fluorescent kind of greenish colors, and those are all shrimp coming up. We do have a native crab. Yes, finally. It is the hardest one to see in the lake though, because it's so good at hiding. These guys actually can be really sizable. So I found one in the lake that was like this big. And it was literally because I was doing something else and was kind of just daydreaming, looking at the water, and all of a sudden the ground got up and then walked away. And I was like, whoa, what the hell is that? The tuberculate pear crab. These guys are really good at coating themselves with things they find on the bottom of Lake Merritt so that they blend right in. This little guy is so darn hardy that we've accidentally exported this to China and Japan. So this is now an invasive species in a lot of their harbors is our tuberculate pear crab. But a really, really cool crab. I haven't seen one though in the last 2 years. It may just be I'm not looking in the right place though. Yellow
Damon,
yep.
we're getting a lot of questions generally about how big things are. So if you could kind of include scale, or I mean not scale, but if you could just kind of for these talk about how big they are, that'd be great. Thank you.
Okay. Yep. So once again, tuberculate pear crab. They can get to be about the size of my head. These guys can get relatively big, and these are definitely the largest crustacean that I find in the lake. Yellow shore crab. If you've ever been on any beach in California that's rocky, you've probably encountered these guys around Lake Merritt. They usually get to be, maybe about the size of a silver dollar. I'm not seeing the same size as I would in other environments in California, but these guys are definitely there. As Lake Merritt has gone through the tidal change of basically having that channel wider and them allowing basically larger tidal influence, which means we're getting more times where land is exposed at the edge of Lake Merritt. I've seen a lot more of these guys. And they'll usually be underneath rocks or behind pieces of wood, things like that, that are exposed on the outside edge of Lake Merritt. And like I said, these guys, Lake Merritt ones I'm seeing are usually about like a silver dollar. Generally, these are like $2 silver dollars kind of stacked together in other environments. This one, when we get into our mollusk side of things, we're now leaving our arthropods and going down a deep dive into the world of soft-bodied, hard-shelled critters. The Asian date clam can be really, really small, up to about a silver dollar size at most. The majority of the ones you're going to see at Lake Merritt, though, are the size of a dime. And I think that's mainly because most of them are generating in a single season and then dying off as we get the fresh water flush in the winter of the lake. You have to remember, Lake Merritt, right? It's got an influence of water from two sides. Salty bay water, fresh water down from the hills. So during the summer, we become much, much more salty. In fact, we have usually, ppms that are very similar to the San Francisco Bay. Sometimes it even gets higher because of evaporation. But then during the winter, we get this giant freshwater flush that makes it more of a brackish water system. And so things like this, I rarely see live ones during the winter, but they reproduce extremely fast. So right now, if you go out to Lake Merritt, there's these giant kind of floating rafts of Ulva intestinalis, this kind of like green seaweedy looking stuff. And it is literally just covered in these, thousands of these in those mats. Most of them right now are about the size of a dime. They're not very, very big. These guys also originally from China, kind of Taiwan, from what we can tell. They also have a very, very thin shell. It's very opalescent. So if you pick up one of these or you find a shell that's already broken open and you can look on the inside, it's like mother of pearl on the inside of them. They're really an attractive looking shell, but very, very delicate, very, very thin, which probably allows them to grow extremely fast. We do have larger mollusks, though, in the lake. We have the soft-shelled clam, and these guys can get to be, about the size of like I don't know, like an iPhone 6 or something. I think is the biggest one I've seen. It can be about yay big. I'd say the majority of these around Lake Merritt are more on the order of like, 10 to 12 centimeters in length. But if you walk around the bottom side of the lake by the amphitheater where we get more of that beach, and you're looking out on the beach, the majority of the white shells that you're seeing out there are this soft-shelled clam. These guys, from what we can tell, probably got imported to Lake Merritt when oysters were first brought to Lake Merritt. And so these kind of probably came over from our east coast originally. Also, if you're walking around Lake Merritt and you're looking in the water on those really clear days and you see this giant puff of like dirt come up in the water, it's usually these guys responding to some sort of stimulus and basically pulling their siphon back down. But easily the biggest mollusk that is easy to find around Lake Merritt are these soft-shelled clams. We also have a number of mussels around the lake, and these are very interesting because they have different dynamics when it comes to saltwater tolerance. So this Atlantic rib mussel does really, really well in brackish water environments where you basically have more and more fresh water. And so there are reports from Jim Carlton when he was doing these surveys way back when that there was a higher proportion of these than when Jim came out here a couple years ago and hit his old basically survey spots, and we are finding a lot of these other species of mussels. These guys are easy to recognize because there are these big black shells. They'll be on the side of Lake Merritt. Most of them are, easily like the size, of your thumb or bigger. They get to be relatively wide as well, but you can easily see the ribbing on these that are running in a radial fashion out the shell. And that's in opposition to these other mussels that we have in Lake Merritt. Don't hold me to this name, to the Middleus edulis, but we do have these things that look more like blue mussels in the lake. They have a smooth shell. They're about the same size as those Atlantic ones, where the majority of them are about the size of your thumb that you're going to find, or a little bit bigger. These are, if these are Middleus edulis in the lake, these are ones we typically, actually people would eat. These guys survive much better in a more marine-like environment, a higher salt concentration. And so we're seeing that it seems that a lot more of these have infiltrated the lake over the years, and that's partly because those Measure DD projects where we're getting more tidal flow, we're getting more of this kind of like oceanification effect on Lake Merritt. If you're interested in learning more about this, Katie Noonan has a great poster on the oceanification of Lake Merritt and can really kind of do a deeper dive into that. Katie Noonan is a high school or former high school teacher who has been basically doing surveys around the lake for decades, and then also doing a lot of the water quality checks and things like that. So that we have this kind of larger picture of what's been going on in the lake. Because sadly, there hasn't been big institutional studies going on in the lake at any sort of high level that would allow us to really see a lot of the dynamics that are taking place. So big props to people like Katie, et cetera, that have been gaining all of that knowledge for us.
Real quick, Damon, we did have a question as you're going through. Which ones are considered invasive? Maybe you can just start from here.
Yeah. So I've been pointing out the ones that are native, which basically means everybody else that I'm not going like, oh my God, a native. Everybody else isn't from here. But remember, invasive has like a weird connotation to it, right? Because it means not only like, is it here, but then it's like taking over space and pushing everybody else out. And so unfortunately, the majority of these ones in the slide deck are what people would consider invasive. In that they are basically pushing out into niches where other locals would usually be. But it's really cryptic to some degree with the San Francisco Bay, which is probably one of the most invaded spaces in the world, because remember, people didn't think that marine invasive species really happened until Jim Carlton came on the scene in the 60s as a high school kid and started asking these questions. And so we don't actually have a really good pulse on where some of these organisms came from. And so that's why you'll hear occasionally the word cryptic applied to them, and that we don't know where they came from, but we're pretty sure they're not from here. And so if you ever run across that this is a cryptic species when talking about being an invasive, that's usually what they're talking about. And we definitely have a handful of cryptic ones in Lake Merritt. One of the most dominant invasive species we have in Lake Merritt is the Japanese bubble snail. Any of you guys that can get out to Lake Merritt in the next like 2 weeks, you will be able to see thousands of these. They blend right in though with the ground. And so they are a mollusk. They have a clear shell that is kind of like this bubble shape, and that's why they have that name. But then their tissue is kind of speckled so that they look a lot like the bottom of Lake Merritt, and they blend in super, super well. These guys are relatively small. So largest ones I find ever are like the size of a quarter. The majority of these, you're talking dime size. But the easiest way to spot them is find some sort of large uniform substrate in Lake Merritt that's been sitting on the bottom, and then just stare at it. And what you'll realize is that all of a sudden you notice that there's like 10 or 15 things moving on it. And it's usually these Japanese bubble snails. Also right now, they're laying eggs like crazy. And so if you look at any kind of this seaweed in Lake Merritt, especially on the, like near the Green Bridge right now, you'll find these clumps that are basically about quarter size that are clear and then have yellow eggs in them. And those are the eggs of the Japanese bubble snail. Getting to the non-shelled mollusks, we get this critter that is just, I mean, biologically amazing. It is one of our natives too, which is also really, really exciting. And triple threat, it is named after somebody born in Oakland. Hedgepeth's sapsucker is named after Joel Hedgepeth, who was born in Oakland in 1911, who was a marine biologist and also an iconoclast. He started what was like the Institute for Non-Progression or something along those lines. Really interesting character. But anyways, this critter is named after him. And the reason it's so cool is for most of you guys that are kind of like marine people, you're like, oh, kind of looks like a nudibranch. Kind of. It's actually not a nudibranch. It's in a different taxonomic group, but it has some really cool tricks. If you notice, it's bright green. It's an animal. They're just using that to blend in. It's using that to actually get food. So this organism can actually steal the chloroplast from the things that it's eating and incorporate it into its body and use those to generate food. So these guys can go up to 8 months without feeding, just generating food based upon sunlight. Another closely related species to this that's on the East Coast takes that trick even further. It doesn't only steal the chloroplast, it steals the chloroplast genomic material and can replicate new chloroplasts with it. I mean, amazing. These guys, I'm starting to see them more frequently at Lake Merritt. I just saw the juveniles out there 2 weeks ago. I would say the last 3 or 4 years, I can consistently find these in Lake Merritt. They love being on this algae that's known as dead man's fingers. Dead man's fingers is kind of this green, thick-looking seaweed, and these guys will usually be crawling on the top of that. Hedgepeth sapsucker can get to be relatively big as far as like a nudibranch-like thing goes. Like this one that you see in the photographs here, this was easy bigger than my thumb. So we're talking like 5 to 8 centimeters, I think, for this one. The majority of the ones I find, though, are more like in the nickel size. And then the juvies are like half a dime size. You really have to know like what you're looking for to find them. But these, I'm finding majority of these are over by the Green Bridge through kind of Lake Chalet seems to be the bandwidth space to find these beauties in. Other mollusks that are really cool that we have in Lake Merritt are these California sea hares. We don't see these every year. We had a really big influx of them 5 years ago, where I think we had a count of over 100. And they had showed up in Lake Merritt to basically lay eggs. And so if you're walking around Lake Merritt and you saw what looked like somebody had spilled a cup of ramen in there, you took a second and started kind of looking around, you'd realize the rocks beside the ramen were moving, and it's all of these sea hares. So they're about the size of a small, rugby ball. They've got some of those cool mollusk tricks, like they could totally spray a bunch of, ink at you and kind of like evade you. They also can incorporate toxins from the things that they're eating and then present that in their tissues so that nobody eats them. We saw a smaller wave the year after that big wave, but I haven't seen them in the last couple years. I've seen them out in front of, I think it was Crab Cove. Me and Ranger James were out there a couple years ago and saw them out there still. So they are still close at hand. They just haven't seemed to come back into Lake Merritt recently. Another cool one that you can find with some regularity in the lake is the sea lettuce eater. This guy, based on its name, you know where to find it. You can find that big, wide sea lettuce. It's usually grooming that stuff. These guys are relatively small. You're talking about 2 centimeters in length. It's got all those projections on its back. That's where it's actually presenting a bunch of the toxins from some of the things it eats so that nobody eats it, because it's just a big soft organism, or a small little soft organism, so very prone to predation. And so its defense mechanism is to present toxins from things that it is eating. We also have other guys in that same kind of ballpark of size of just a couple centimeters. Placidia dendritica is seen a couple times throughout the year at Lake Merritt. We also have another organism named after Hedgepeth, and this is definitely one of the showier kind of nudibranchs in Lake Merritt, which is, Polycera hedgepethii. So Polycera, poly refers to many, Cera refers to those projections that are on this organism where it is presenting the toxins from the things that it is eating. It also, of course, warns predators about this by basically lighting those up with bright colors, yellows and blue shades. These guys are also relatively small, so we're talking just a couple centimeters. Constance, are you drawing on there, or is that somebody else?
That is not me. I thought that was you. Interesting.
No, that is not me. If somebody was drawing, is there a chance you could clear that? Otherwise, we'll just go with what we've got. Okay. Oh, thank you. Santa Barbara Janilis. We saw this just a couple years ago, which is actually really, really surprising. This is a really large-sized nudibranch that you will see probably only a handful of times in Lake Merritt. This is something you usually would see on the open Pacific side of our coastline. This guy can actually, or this guy, gal, she, it, can get to be relatively large. So these ones I literally saw while walking around the edge of the lake over towards the Glen Echo watershed input. This specific one I photographed, I think, was on the order of 5 to 7 centimeters in length. Also does a similar thing, projects toxins out into those outer sera, and then brightly labels them to tell things, hey, don't eat me. We also have the Lake Merritt nudibranch, which I have never seen. I still need to see this thing. This is a cryptic species in that we're pretty sure it's not from Lake Merritt, but Lake Merritt was the first place it was seen at. This was photographed by Kenichi. So this is one that's still on my to-do list. We have a lot of the eastern mud snails in the lake. This is the mollusk that you can see kind of throughout the year in Lake Merritt because it survives in the very low salinity and high salinity times. These guys can be relatively good size. They're about the size of an average garden snail. So you can definitely see these as you're walking around Lake Merritt and just kind of looking down into the lake. They're usually pretty darn dirty looking. Uh-oh. Let me go back to screen share.
Sorry about that. Hang on one second. Uh-oh.
Are we back to screen share?
Oh, did you get it back?
Yeah, I think so.
Okay. Sorry about that.
That's okay. And so these guys you should be able to find just about every location in the lake at all times of year. In fact, the easiest time to see them is probably during the winter because there's nothing else competing with seeing them at that point. And they'll leave these long kind of streak marks in the lake. These guys also non-native, invasive. Originally, we got these when oyster beds came into Lake Merritt from the East Coast. Uh-oh. Okay, there we go. We also get limpets in the lake. Limpets are kind of a cool mollusk if you haven't seen them before. They're a single shell and they can spend a lot of time out of the water. And so if you look on the outside of that shell, it's got these little small projections. So we can use those projections to more or less do air exchange when it's out of the water for hours upon hours. And then when it's in the water, you can see that kind of like feather boa sticking out from beside the head. That's its gill. So we can use that then to do a more efficient exchange of gases when it's under the water. So these guys are usually grooming hard surfaces in the lake, just kind of mowing over that little algae layer that comes up on rocks and pieces of the dock, things like that. These guys, for the most part, I just saw my largest one in Lake Merritt just last weekend, and it was just barely bigger than a quarter. So they're not huge, but they're big enough to easily spot with your eyes if you're just kind of paying attention. Another invasive species we picked up from those oyster beds coming in from the East Coast is the eastern oyster drill snail. This is the nemesis of all the other hard-shelled organisms of the lake. And that's because this thing eats them. What it will do is it'll roll up on another shell, and it has kind of a strong kind of rasping-like device inside that foot that it can use to basically drill a hole into a shell, and then it can spew in digestive enzymes and then slurp those back out and eat them. So if you check shells on the beach at Lake Merritt, especially those Asian date shells, because they're so small and so thin, you'll find many of those that have these perfectly little round holes in them, and that is from this little guy here, the eastern oyster drill snail. These guys also can be about the size of a quarter. The majority of them, though, are about the size of a nickel. This is the eggs from them, which is also really, really common to see at Lake Merritt. These eggs are relatively small. We're talking probably 1/4 of a centimeter for those ones that you see there, the individual egg stacks. But you'll see these large clusters of them on pieces of seaweed on the bottom side of rocks and things like that. And this kind of odd vase shape makes them really easy to identify. They're also really brightly UV fluorescent. So if you're UV fluorescing them, hitting the lake at night, you also will find these with great ease. We have a organism that I actually worked on at the National Labs, where we were actually trying to figure out how these things break down wood because they're so good at it, which are these things called shipworms. Total misnomer when it comes to this, to the common name. And that's why scientists love Latin names. So what we think we have in the lake is Teredo nivalis. To actually identify that, we'd have to look at the small platelets on the back, which I've never been able to recover on one of the ones from Lake Merritt. But these are a really interesting mollusk in that they have this worm-like shape to them, but their head is basically where the shell is. And they use that shell to literally drill into wood, basically scrape it up into small particles, and then symbionts that live inside of their gills exude cellulases out into the tube they're drilling in, break down the cellulose in the wood, and then suck that back down into their GI, where they've got another set of microbes that actually can further take that degraded wood now and turn it into sugars for this organism to live. So if you accidentally break open a piece of wood at Lake Merritt and you see these calcified tunnels in there, it's most likely you're seeing shipworms. These are also the whole critter why Columbus didn't get back to Europe on his arrived time, the third trip, because these completely ate out his ship. So they really do a number on wood. Between these and that isopod from New Zealand, Australia, like a chunk of wood in Lake Merritt has a pretty long or pretty short life. We also have some of these dynamic looking flatworms in Lake Merritt. I still have not been able to get this further than a suborder. I apologize. But these guys you'll find predominantly under rocks at Lake Merritt. So if you pull out a rock and then set that in a pan, you'll see these guys kind of immediately like float off of it. These are also, we're talking quarter size. Some of them will get larger. I found one that was about the size of a half silver dollar piece in Lake Merritt. What you see on the ventral side there is the gonads, so that kind of white aggregation. But these guys are paper thin. They just have little tiny eye spots, and then the gonads, and that's about the only recognizable things that's easy to see without getting microscopic with them. We also have a ton of Cnidarians in Lake Merritt. When I say Cnidarians, most people recognize these as sea anemones and jellyfish-like things. So we have the orange-striped green sea anemone, which is a known invasive species from China and Japan. These guys are in great numbers right now at the lake. They're relatively small though. So with their tentacles out and exposed, you're looking at something the size of a quarter. They're not the huge green sea anemones you might be used to from the California coast, but they are kind of aggregating on all of the hard surfaces right now. And sometimes, like summers like this, I'm even seeing them attach onto some of the developing kind of seaweeds and sea lettuce out there. And so if you basically just kind of spend your time looking down at some of these hard surfaces, I can almost guarantee you that you'll find a handful of these out there. Here's a more side shot of them so that we can kind of get a feel for the size of them. This one was on a piece of sea lettuce a couple years ago. We also have a handful of jellyfish in the lake. So we have Pacific sea nettle occasionally makes its way into the lake. These guys are a really showy jellyfish that you'll usually see more out in the San Francisco Bay or even sometimes on the coast. We also get bell medusas in the lake. This is my first year being able to catch one and photograph it. These guys can actually get to be, you know, a decent size. Like they'll be about like half the size of like a pop bottle, like a soda can. And you'll find them at the surface just kind of like bobbing up and down. When we found this, we ended up finding, I think, 3 of them that day. And so they kind of come in big waves into Lake Merritt. They're probably coming from the San Francisco Bay and coming in during an in tide. We also get the big moon jellyfish that if you've been in the bay, you've probably seen. These will also occasionally make their way into Lake Merritt. Usually not large numbers though. It's usually just a one-off. We also have gooseberries that make their way into the lake occasionally. I found these this year. Super, super cool. These are the ones that like are so beautiful that you'll find, you know, Monterey Bay Aquarium basically displaying other ones within this group, the cenophores. That's because they've got these ciliated outsides that if you hit them with light, it literally kind of reflects back a rainbow up and down them. These guys are about the size of like a golf ball at the largest. These specific ones that I have photographed here, these were more on the order of like a quarter, if you were to take a quarter and make it spherical, about that size. Now moving away from all the Cnidarians, the jellyfish-like stuff, let's talk about one of the really diverse groups in the lake. Actually so diverse that people like myself that are pulling stuff out of there get asked by groups if we can send them samples because there are people interested in studying these, as we think that we might have minor evolutionary events taking place in the Lake Merritt or just species that haven't been documented before. So this is the family or the group known as the Polychaetes. This kind of stacks under your annelids, your segmented worms. And so in common speak, these are our marine segmented worms. The clam worm or these Aletas can get to be really large. And so I think the largest one I've pulled out, I think was probably, hold on one second. Always also helps to have a measuring device around. Yeah, the largest one I've seen out of Lake Merritt was probably about somewhere between 25 and 30 centimeters long. The majority of the ones I find of this tend to be on the order of 15 centimeters. So 15 centimeters, for example, about that big. These guys are usually hiding underneath rocks or in kind of piles of mussels. For the most part, these guys are eating other stuff that have decayed. The one thing is you do have to watch out for on these is they do have a really large set of jaws. I really need to get the photograph of this in there. But I mean, if you look at them, they've got these really large calcified chompers, which for the most part, never bite anybody. Allison over at Cal Academy, who's handled these for, eons, has never been bit until I think last year by one. So your chance of actually handling one of these and getting bit is relatively low, but it's something to be aware of if you're interested in manhandling one of those. One of the ones that is really cool in the lake, and we think, going back, we think this one is probably native. There's some variations on this one though, and this is why LA County Museum of Natural History has asked me to send a handful of these down there, is that they think there might actually be a different species here, one that hasn't been identified. So we've sent them samples the last couple of years, and they're working that up using some DNA sequencing, along with morphological looks at kind of the size of those antenna-like things coming off of the head. One of the ones that was actually identified by LA County way back in the day, Leslie Harris, who's basically a world expert in polychaetes, was, I think, the first person to identify this in the San Francisco Bay, is this really beautiful polychaete from Japan, Megacillus niponica. And this one is relatively small. It's a relatively thin little thread. And so you could think about it as being about as thin as like half of your normal like shoelace. It's really small in that regards. They get to be about, I think the largest one I found has been maybe 7 centimeters. They're typically more down on this scale of like 3 to 4 centimeters, but they're really easy to recognize down to a species because they have this dark band around their kind of thorax, not thorax. Oh, I'm blanking on the name. Pharyngeal area. Up towards the top of their head, there's this really dark band that makes them easy to recognize. In this photograph, you can see the bottom of this one looks like somebody kind of squished it a little bit. Nobody squished it. What's happening there is we're starting to see the beginning stages of how this thing reproduces. You gotta remember, most other organisms reproduce in a much more exciting way than humans do, and this is definitely a highlight one. So what happens is at a certain point of the year, actually starting right now, these guys start to send all of their gonad material down to the bottom side of the body, aggregate it down there, and then that bottom side of the body starts to bud off. It actually buds off so much that it becomes this thing. It changes color, it gets its own eye spots, and it goes swimming by itself. It's basically like if your genitalia decided to kind of like move its way into your leg, and then your leg was like, see ya, and like broke off, got some eyes, and like walked down the street. They basically will swim around, find other groups of these, and these are known as epitopes, this life cycle stage. And they'll find other ones of these until there's enough of them, and then they'll more or less kind of lyse open and intermix the reproductive eggs and sperm. The reason that an organism might use a strategy is that the adult doesn't want to put itself at risk to go out and find other partners. And so the adult can basically stay hidden under a rock or in some mussels and then just bud off an epitope and let that go do the reproductive stage and stays totally safe. It doesn't have to worry about those birds and other things eating it. And so right now you can start to see these, and this is the earliest I've ever seen them. Usually it's September I see these, but I just picked up my first one last week at the end of Madison Street. You'll see these little tiny orange things like swimming really rapidly and erratically at the surface of Lake Merritt, and it's the epitopes from this Megacilus niponica. Some of the other polychaetes in Lake Merritt will use a strategy. Not all polychaetes use this strategy to reproduce, though. Another one that does use it, though, is this beautiful looking polychaete from New Zealand. We found this in 2015 with Oakland High students. I was actually just supporting Katie Noonan and something. This was the first time that this was seen in the Bay Area. The closest other observation was in Ventura Harbor. And then outside of that, the only place else it's been seen is basically New Zealand and Australia. Really, really small though. You can see in the bottom picture there's a dime, and this thing is like maybe taking up the print type of Liberty in the dime. It's really, really tiny. But the backside of it, you can see all those epitopes starting to bud off. We also have other types of polychaetes in the lake that don't do the epitope thing. This is a spaghetti worm, so it basically hides usually in a mud hole and then has all these tentacles that it uses to grab things around it and then pull it back to its feeding tube or its feeding hole. Another type of spaghetti worm looking thing is these Terribilides that we have in Lake Merritt, which are really pretty if you ever get a look at them. These are usually encased in mud, and so they're usually not this pretty. This one I took out, washed the mud off so that you could see the setae, the little segment pieces sticking out. And then also you can see its air exchange system. Those kind of branching, bloody looking things are how it's basically doing oxygen and carbon dioxide exchange. Other polychaetes we have are these ones that kind of play a similar trick as butterflies do a lot of times. They have these scales on the outside that then have shapes that look like eyeballs, hopefully to scare predators off. So we have a lot of these 15 scaled worms in Lake Merritt. These guys are usually occupying the size of like, up to about 5 centimeters. I haven't found any really big ones, and I usually don't find any really small ones. They're usually all in this kind of same ballpark of 5 centimeters. This is also a native, yay! These guys you'll find usually in clumps of mussels. We also have other kind of crazy looking polychaetes that will dig into the actual mud of Lake Merritt, send out tentacles and grab stuff. I haven't been able to get any of these past the genus level, and I don't even know, to be honest, I don't even feel comfortable at the genus level call of this one. We've got other polychaetes that are more kind of predatory roamers that are out there, like this Merphasia that you'll occasionally see. I don't see this one a whole lot. This one was actually relatively large. This was more on the order of like 10 to 15 centimeters. So pretty good sized polychaete. One of the most important polychaetes of the lake, though, are these ones that produce these large calcium encrusted tunnels on substrates in Lake Merritt. This is Phacomopolis enigmatis. This is one of the first organisms at Lake Merritt that we realized things were getting here from Australia. So Jim Carlton was really, this was a species that he was very, very well with, did lots of documentation on early on. These guys, for the most part, don't cause problems in Lake Merritt, but in Southern California, sometimes these will make so many of these calcium carbonate tunnels that they become reefs that actually are blocking harbors. So I think Ventura's actually had to go through and like dynamite a couple of these reefs from these guys. But these are really cool if you watch them. They basically send out little graspers that will then feed on stuff coming by in the water. And if you get too close, they'll pull back in really quick. So if you guys are familiar with like Christmas tree worms, this is basically our Christmas tree worm of Lake Merritt. We have other tube worms in the lake that I have not been able to take down to any sort of taxonomic certainty, but they are there. We also have bryozoans in the lake. So this is kind of the group, I would say, most closely related to like coral-like things. So they're colonizing organisms. They'll usually produce a calcium carbonate kind of structure that they all live in. Each one of those little holes you see there basically has an individual that can come out and feed. A lot of these guys produce pretty gross toxins, and that's why nobody tries to feed on them, except for some of those mollusks who try to feed on them and then present those toxins to other organisms trying to eat them. These guys are usually encrusting organisms. And so each one of those little tiny like keyholes you see is like really, really small. That's being taken with a macro lens. But most of these aggregating colonies that you can find will be about, quarter to silver dollar size. And it takes a little bit of time to recognize them. So what you want to do is like pull up a rock or a mussel or a clump of mussels and look at it and look for a textured pattern. Sometimes you have to clean it off first, and then you'll recognize that you're seeing these there. A little small hand magnifying lens does a lot for being able to really get in close and see these guys for their full beauty. Another type of bryozoan we have that people often think is a plant when I show it to them are these bugula species. And that's because they grow into these large kind of feathery clumps that look much more like a piece of seaweed than it does an animal, but they are just kind of like a coral, a giant aggregation of a whole bunch of individuals. And if you get really, really close on this image, which you're not going to be able to do on Zoom, but if you've got the slide deck there, you can actually see on one of these, you see a little feeder coming out of one of those ones on the top. And so they are also there just kind of filter feeding the water. As far as I know, this is an invasive species. We're not going to, I don't have a whole bunch of chordates in here because I think they're really exciting stuff for all those weird invertebrates we just went through, but just to highlight it, because I know for people, they really don't think a place is amazing until they see like a recognizable large organism like a fish. So we're gonna do a couple of those just to remind you, those are in there. Bay pipefish, everywhere in there. Like we find them when we pull stuff from the boat dock. We find them when we pull stuff from the Green Bridge. When like you go under Lake Chalet, they're there. They're just kind of hard to see because they're so good at just kind of blending in. They'll just kind of hang out in some of that seaweed and just hope you go away. But if you pull out some of that seaweed and you kind of put it in a pan a lot of times, they'll all of a sudden be like, oh, my environment's changing, I'm gonna have to swim away, and that's usually when I see them. These guys can get to be pretty long. I think the longest one I found in Lake Merritt was pushing probably about 15 centimeters. So they can get to be really good size. The majority of ones I find though tend to be more on like that 10 to 12 centimeter size. I'm sure the little ones are there, but I just can't see them. We also have a ton of yellowfin gobies in the lake. These guys are non-native, originally from Australia, where our pipefish are native. It's awesome to see those guys in there. Yellowfin gobies, though, are just freaking everywhere. When you're walking around the lake and the majority of fish that you see all of a sudden like shuttle off when you walk around, it's usually these guys. If you flip over a rock that's in the mud when the tide's down, 50% of the time you're going to find one of these guys underneath there. They get to be also, decent size. Like they'll get up to be about, 10 centimeters, but they're nice and like chunky. They're not like the pipefish, which can be really thin. So these guys are pretty hard to miss if you're actually going out and looking for them, and just in inordinate numbers of them. And this is also one of the ones I like to show just because it's like one of the things that gives hope to people. If you go onto YouTube and you type in Lake Merritt bat ray, you'll be able to see it. I'm not going to try and show it here just because internet connection's crappy, and so like you guys will just see a glitchy video. But you can type that in and hit play, and you can see this is taken from the Green Bridge, where you'll often see the bat rays feeding. So during tidal change, they'll come there and basically be feeding on stuff that's kind of moving in and out of that tidal system right there at the Green Gates. We had a big die-off a couple years ago, just like a lot of sharks' rays had die-offs in the bay. That was still being investigated. They think it was a protozoan due to, I think, increased temperatures in the water. I have not seen bat rays in there this year. We do have a ton of other fish in the lake that I'm not going to highlight just for the sake of time. We have striped bass in there all the time. When I first moved to Oakland, I'd walk around the lake like at night, like in early 2000s. Used to be a couple of like old guys that'd be hanging out there like with their full-on fishing vests, catching stripers like nobody's business. And you can still see those stripers in the Lake Merritt relatively frequently if you hang out in this same space down there by the Green Bridge. They'll usually duck into the shadows and then come into the light to grab stuff and then move back in the shadows. So it might take a while for you to see stuff. How can you learn more about all the other things there? Because like we covered a lot and you guys are very generous with your time. Sorry about going over. there's this platform that you might not be aware of that I'm a total addict on. It's called iNaturalist. It's a platform where any user using their smartphone or their camera can take pictures of things and upload it to a global database where people help identify what that organism is. And in the process, you end up building this entirely rich map of species biogeography. In fact, it's amazing to see what this platform has done just in the handful of years that it's been available. So for Lake Merritt right now, we have over 10,000 observation of different things there. You can take a picture of any living thing and upload it there, and somebody may be able to help you identify it. Right now, we're at about over 1,000 species at Lake Merritt, which is pretty impressive. But you know what? It's just the frickin' tip of the iceberg. There's another 97% of that world in Lake Merritt that needs to be documented for sure. Other citizen science things going on around Lake Merritt. This has kind of come to a standstill just due to problems with the space we are using, but I will try to get this back up and running as soon as I have lifetime again to do it and a functional Rotary Nature Center. We had a Barcode the Lake Merritt project going for a while with the goal of basically getting DNA sequences for these different organisms around Lake Merritt. The reason this came online was because people were asking for samples so that they could do DNA sequencing to figure out these things were for like polychaetes and stuff. And I thought, I know how to do this. I would love to make this something where locals can come in and learn these techniques, generate the data, and we build this really, really rich data set of what lives around Lake Merritt, not just photographic information, but also basically DNA information that can allow us help parse some of this taxonomy that's a little dubious. We started mainly with fungi at Lake Merritt just because that's where my strengths are. But the goal is to eventually move into all that marine stuff, especially those polychaetes. I got my eye on those for sure. You can find Barcode the Lake by just typing that into Google. It's the first thing that comes up. And hopefully once I get some personal things squared away and RNC gets up and running again, we can get this project off the ground again. That's all I've got for you guys. Now the Q&A.
Wow, thanks, Damon. That was fantastic. Ooh, a whirlwind. We do have a whole number of questions. It is, let's see, quarter after. Maybe we can just do a couple. And as people are kind of inevitably going to be trickling out, I am going to share the donation websites again, as far as places to donate while we are going through a lot of structural change in our country right now. And yeah, so we've got those, and then I've also included the slide deck for Damon's presentation, as well as the link that Katie sent that there's a lot of great information about Lake Merritt there as well. This is being recorded, so we will post it on the CCNH website and send out an email when that happens. So we had a couple questions that I think are kind of more general interest, as far as what's the story with paddling your own boat around Lake Merritt? And then a second question is, how do you get all of your amazing pictures?
All right, so the first thing, paddling a boat around Lake Merritt, you do have to pay a launch fee. So it's a $5 fee to launch your boat, which helps basically pay for a lot of things that you see going on around Lake Merritt. You can also rent boats directly from the boating center at Lake Merritt. They have a nice diverse portfolio of things you can rent. There's educational programs about how to use the different vessels. So I'd say inquire there, and they have a lot of offerings there.
Awesome. And then another question was, how do you take your photographs, and also how do you find the organisms?
Okay, so finding the organisms, it just takes patience, is usually what it comes down to. So when I first started finding organisms down at Lake Merritt, it was because of that walk with Joel Peters talking about measured ED. And so as soon as that walk was over, I literally just went down to Lake Merritt and just stood there like this, looking at the mud. The first thing I saw was a shell, and I picked that up, and I realized that shell had something on it. And then I started looking at that, and you just say kind of branches out, right? So The best thing to do is find hard substrates, though. If you're looking for a diversity of things, find rocks in the lake. The underside of docks or the side of some of those pillars in the lake are really good. Or even just where Lake Merritt has that cement wall, a lot of organisms are living right where that cement wall hits kind of the muddy bottom of Lake Merritt. And so those are where I'm finding like 99% of all the organisms you guys saw pictures of are literally probably like 2 or 3 feet off of shore at most, mainly on hard substrates. Most of those photographs I'm taking by taking the organism home in a piece of trash that I find at Lake Merritt, putting it in a cereal bowl, and then basically working my photography magic. I've got just a DSLR with a macro lens, and then a lot of times like I'm spending like an hour to try and capture an image that illustrates what that organism is, and then quickly returning that organism back to Lake Merritt, so that I'm not damaging anybody in the process. But full disclosure, there has been fatalities in that process. Luckily, nobody that was an irregular. I did have a couple Japanese bubble snails that didn't make it.
Somebody else was asking about, are there paddle tours of the natural history of Lake Merritt? We used to do a lot of walking tours with CCNH. We have never done a paddle tour, though. To my knowledge, nobody is really regularly doing that. I don't know if anybody wants to comment in the chats about if that is, anything that might be going on with that, then please go for it. All right, another question, a couple kind of organism questions. Do the sponges grow in a particular region of the lake or all over? And how big is really big for these sponges?
So the sponges grow all over. So I mean, like the ones I found 2 weeks ago, that when I say relatively large, I mean they were like, the size of like a soda can worth of sponge. And those were just by where Madison hits Lakeside Drive. I know being with Katie before over the boat docks, like they find them all the time over the boat docks. I find them all the time down at the Green Bridge, and I find them all the time at the east side. So they're kind of like all over the lake. But once again, find hard substrates is the best and easiest way to find those sponges.
Great. We had another question about, are there Daphnia in the lake?
Not that I had seen. Remember the lake is brackish water, and I'm not sure how well some of those Daphnia would do in that. I've not seen them in there. Katie Noonan would be the one that, if they were there, would have seen them because of all of the work that her and her students have done looking at organisms, zooplankton in the lake.
Cool. Katie just put something in the comments. Rotary Nature Friends has sponsored 3 walks at the lake since last October. We also have had ROV tours, which are the underwater cameras from the pontoon boats out of the Lake Merritt Boating Center. And we are due for one this summer. Excellent. Here's one. When you observe something, what is your approach to identification? INAT or something else?
So I usually start by, my first thing is photograph what makes that thing different, right? And then get that on INAT as fast as possible. And then I'll start basically looking through observations in INAT of the San Francisco Bay. Because a good chance that I'm not finding like a new thing that nobody's seen before. So there's a good chance that organism's probably been spotted in San Francisco Bay before. If I strike out there, then I pull out Waste Manual, which is like the Bible of like marine biology for the West Coast. And I'll basically start going through the key there to see if I can kind of get it down the taxonomical ladder a bit. And then once I've done that, I'll readjust my iNaturalist observation for that, which helps then anybody that is an expert in that, that's basically hanging out on iNaturalist and looking at their favorite group of organisms, to be able to find that easier and help me identify it further. I mean, because you got to remember, like, my background is I'm a molecular biologist. I don't have an academic training in any of this. So a lot of this I've learned on the fly. And things like iNaturalist and just community groups has been how I've picked up most of my knowledge about these things. So these are all things we can pick up if you're interested enough.
All right, maybe one or two more questions. One of them is, when can we start installing better habitat structures for the invertebrates, e.g. wetland and subsurface structures?
That's a question beyond my my pay grade. That's one that's going to come back to, I would say, the people that were instrumental on Measure DD would probably be the ones that could work on something of that level. I mean, we do have a fair number of substrates that make their way into the lake. They're just not always intentional. But it would be cool to see some intentional habitats in Lake Merritt for those organisms.
Cool, and maybe one more. Do you know of any major differences, or the actual question is, what are the major differences in the flora and fauna since Lake Merritt was reopened to the tide?
Well, it's hard because there wasn't an academic group studying this. So we kind of have, a composite of different little things. Katie Noonan has some of the best data on this. And then also Andy over at Tiburon had been doing plates and studies. And so it's basically usually contrasting that data against what Jim Carlton had been doing in the 60s. And I think his stuff stopped in the 70s. And that's about the granularity that we have. So I mean, a lot of it literally is coming through, I would say, is this kind of citizen science of people observing stuff, documenting it into areas where other people can see that data and trying to kind of compile what's going on. And that's why I can refer you back to this poster that Katie had on the oceanification of Lake Merritt that she might be able to share maybe in the chat, or you can follow up with her about it later. But it kind of gives us a glimpse into these changes that are taking place. I don't have any like great hard line, like this species right here, that was definitely Measure DD. All I can tell you is at a qualitative level, I'm finding a larger diversity than I had when I moved to Oakland in 2001.
Great. There are a lot more questions, but we've been doing this for about an hour and a half. So maybe what we can do is we've been keeping track of all the questions. We had a request to keep the chat as well and send that out to participants. And yeah, we'll send out an email to let folks know. I think we can even send out an email to Eventbrite, right? So if you're not on our email list, then people who signed up for this program can still get the chat and a copy of the recording and all of that stuff. Great. And then maybe one more time, I'll just put in the links to the organizations that folks can donate to that are supporting all the actions happening both tonight and around Oakland and the larger country. So yeah, I wanted to say one last thing, that the things that are causing a lot of our environmental destruction and species loss are intimately tied up with structural racism. So these things are completely interconnected. So as we are watching all the Black Lives Matter movements and everything that's unfolding in the news, yeah, it's important to keep in mind that all these things are connected. So yeah, I'd like to thank all the people who are protesting and all the people who are working to make a change in the world. And yeah, Black Lives Matter, and that's all I have to say.